You're tuned into Human Anatomy and Physiology. Today we're going to be discussing some excerpts from Chapter 3 on cell biology, some subtopics that will help us in our understanding of the various tissues and organs as we look at the organ systems of the body. Cell theory states that cells are the smallest living thing, that everything that we consider alive is made up of cells. Cells do not arise spontaneously, they arise from existing cells by cell division. There's a lot of different types of cells, 200 different types of cells approximately in the human body, and they differ because of the proteins that they express. Every cell has the same genome or set of codes or recipes for making proteins, but every cell type expresses a different subset of those proteins, and it's that subset of proteins that gives the cell its properties, determines its size and shape, and what types of organelles it has. Here's just a quick little collection of cartoons that represent some of the different cell types in your body, most of which I think you'll immediately recognize from experiences you've had in studying the cell. So again, all of the cells have the same chromosomes in your body, and the chromosomes are collections of recipes, if you will, codes for synthesizing specific proteins. Each cell expresses a specific set of proteins that give it its properties. And here's a little review of kind of what's going on with respect to protein synthesis in cells. Transcription factors are proteins that control the expression of genes. That means for a given recipe for a particular protein, there are proteins, other types of proteins called transcription factors that determine whether or not that recipe will be utilized to actually make proteins. So what happens? A transcription factor binds to the promoter of a gene. The gene is then transcribed into messenger RNA, so that the pink ribbons represent the double-stranded DNA in all your chromosomes. We copy out the recipe for the protein, the sequence, into a messenger RNA, which is then transported from the nucleus of the cell out into the cytoplasm and then used by ribosomes, little machines, to actually synthesize the proteins. And here we can see a brand new nascent protein arising from a ribosome that's reading essentially the code, the sequence of the messenger RNA. Cells keep on producing new proteins and constantly degrade old proteins. They never stay a long time in the cell. They're constantly replaced and renewed. But we can control the expression of various genes which determines all the proteins that will be found in a given cell. So let's look quickly just at the structure of a cell. We're not going to look at all the organelles in detail. I encourage you to read Chapter 3 through if you're not familiar with cell biology and you don't remember much about maybe, say, a cell biology course that you have taken, or if you haven't taken any. But I just want to focus on a couple things, and here's a list of components of a cell that I think are worth remembering what they're doing. The plasma membrane is an outer seal, a boundary around the cell that allows it to control its internal environment, as distinct from the outside. The cytoplasm is the liquid, the water that's entrapped within the membrane of a cell, and within the cytoplasm are many, many proteins and organelles, structures within the cell that have particular functions. The cytoskeleton, a lot of times people come up with an understanding of cells, understanding they have a boundary, a plasma membrane, and understanding there are some organelles inside. But the plasma membrane doesn't really contribute to the shape or integrity of a cell. It's just the seal. The cytoskeleton is a weave or scaffolding of proteins within the cytoplasm that actually gives the cell its structural integrity, its build. And it also may determine its shape. So we'll take a look at some aspects of the cytoskeleton. The nucleus is the place in the cell where all of those recipes, all the DNA, the chromosomes reside that contain all the sequences of the information for producing proteins. As we saw, the DNA then would be transcribed into messenger RNA when activated, and the messenger RNA would be shipped out in the cytoplasm to allow us to synthesize proteins. Cells are chock full of organelles and cytoskeletal structures. Here's a cartoon attempting to represent the nucleus of a cell surrounded by endoplasmic reticulum, which is related to the processing of newly synthesized proteins. The Golgi apparatus, which is involved in sorting and packaging and shipping proteins, either to be destined for outside the cell or for functions within the cell. We also see some mitochondria. Those are the energy producing factories of the cell. They churn out a ton of ATP, the chemical form of energy storage, and cells use ATP for virtually all activities and functions. Here we see some centrioles that are important in cell division. We can even see emanating from the centrioles some microtubules, and we'll talk more about the microtubules when we talk about the cytoskeleton. So the plasma membrane, it's that barrier, that boundary that separates the cytoplasm and its solutes and components from the extracellular fluid. So that's how cells can maintain homeostasis. There's this boundary between the two. The cell membrane is actually composed of phospholipids. Those are the chemical elements, building units of the lipid bilayer of a plasma membrane. They have a water-loving or hydrophilic component, which faces the water on the inside or outside of the cell, as we'll see in a moment in a cartoon. And they have a lipid-loving interior component that will be interesting in that it controls what can cross over the cell membrane. The lipid bilayer is not the only part of the cell membrane. There are many membrane proteins, and we'll wait to talk about those more until we have a nice diagram to be able to visualize what in the world's going on with those proteins. So here's a little cartoon that shows the lipid bilayer. Two layers of phospholipids, with the yellow spheres represent the water associating part of the phospholipids, and the little squiggly lines represent the lipid loving or oily tails that find their way to the inside of the cell membrane and determine what can cross. The only thing that can pass through this lipidy interior are fat soluble or lipid soluble substances.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions such as sodium ions and hydrogen ions cannot pass through the membrane unaided.
So unaided. Here's another cartoon, a little more complex cartoon, showing the same thing, showing the phospholipid bilayer that represents the seal, the boundary on the outside of a cell, and it has a lot of membrane proteins associated with it, some of which span the membrane from outside to inside and vice versa, and those are called transmembrane proteins, and they have a lot of functions that they contribute to the cell. Some of the proteins are associated with the inner surface of the membrane. They're called peripheral membrane proteins. Let's take a look at some general functions, categories of functions of membrane proteins. Some of the transmembrane proteins are the things that aid. Our narrator indicated that glucose and amino acids and ions and so forth cannot cross the lipid bilayer unaided. Well, there are proteins that can shuttle those substances across the membrane. They're called transporters or transport proteins. Some of them passively transport substances across the membrane, as we'll see, and some of them use ATP to pump, if you will, substances across the membrane. Some membrane proteins are receptors for chemical signals. Cells have no eyes, and the way they can determine kind of what's going on around them is by specific chemicals that bind to receptors on their surface, and the resulting effect is that changes occur within the cell. The membrane protein will then change its function after binding to its ligand or its signaling chemical, and signals will occur inside the cell behavior will change. Some membrane proteins have the role of attaching the cell to fibrous proteins that reside on the outside. The extracellular matrix, the substances, the material that's outside of the cells, contains a lot of fibrous structural proteins, and the cells are typically anchored by membrane proteins to that extracellular matrix. Here's a more complex slide, or a cartoon I should say, showing a plasma membrane with some transmembrane proteins that anchor the cell to these fibrous proteins in the extracellular matrix. In addition, the transmembrane proteins anchor themselves to the cytoskeleton. There's a complex scaffolding of proteins inside the cell, which gives it its integrity, and the membrane proteins attach the cytoskeleton to the extracellular matrix. That's what gives the cell some integrity and gives it some location. The lipid bilayer is purely a boundary. It's not anything to do with the structural integrity of the cell. Some membrane proteins are enzymes. Sometimes when a membrane receptor becomes activated, it will in turn then turn on an enzyme inside the cell, which will produce some product, which will in turn change the function of the cell. So that's one of the ways that membrane receptors can change the cellular behaviors, by activating enzymes that are in the membrane. There's a little cartoon showing an enzyme inside the cell. Some enzymes are associated with the membrane, some are freely floating in the cell. Some enzymes reside inside of various organelles.
An enzyme is a protein that changes the rate of a chemical reaction without itself being changed into a different molecule in the process. Enzymes promote and regulate virtually all chemical reactions in cells.
So we'll talk a lot more about enzymes in the context of each specific tissue type and its function as we go through our text, our narrative. Other membrane proteins allow cells to recognize other specific cell types. Cells of your immune system, for example, are able to recognize specific cell characteristics based on their membrane proteins. So here we see two cells with cell-cell recognition taking place because one membrane in the cytoplasm, or in the membrane of one cell has a specific recognition capability for this unique protein on the neighboring cell. We'll talk more about that when we talk about the immune system. Most cells have cell-cell junctions. So here we see a simple representation of two membrane proteins that anchor or attach to each other, kind of like Velcro. And cells are often stitched together by cell adhesion molecules, or cell-cell junctions. And we'll talk about those in a second. So a lot of functions of membrane proteins depends on how they're structured and to determine what their specific role is. Let's now take a look at three different types of cell-cell junctions. Most cells are, as we said, attached to other cells by cell junctions. In fact, some cells are attached to the extracellular matrix, or most of the time by cell junctions. These three are cell-cell junctions, tight junctions, desmosomes, and gap junctions. Here's a sheet of cells, or part of a sheet of cells. They're epithelial cells, as we'll discover in Chapter 4. They form boundaries from one part of your body to another, or for one compartment of an organ to another. And the way they can form boundaries is because they're kind of zipped together, stitched together, if you will, by tight junctions. That controls or limits what can pass between the cells, and that allows the cells to form form effective boundaries from whatever's outside this epithelium and whatever's inside. So oftentimes when one side of an epithelium will face the outside world, while the other side of the epithelium will face the inside of the body, and we want to be able to control differences between those two compartments or regions. Desmosomes are cell-cell junctions that are designed to tightly attach the cytoskeleton of one cell to the cytoskeleton of another cell. Right? The tight junctions are more about a seal, about the boundary between outside and inside of a boundary layer of cells. The tight junctions are mechanical connections like snaps or buttons between the cells, or Velcro. I like that analogy. So desmosomes anchor the cytoskeleton of one cell to another. Remember, it's not the membrane that gives the cell its attaching integrity, it's the cytoskeleton. And so the membrane here is the seal. If I can just get my mouse going here, and the cytoskeleton of each cell needs to be anchored via desmosomes. And finally, gap junctions. Gap junctions are like little pores in the membrane of one cell that attach or associate with like pores in the neighboring cell, forming an open passageway so that ions can diffuse readily from one cell into another. We'll see what makes things diffuse in just a bit. So there are three types of cell junctions that most cells have. Some cells don't have gap junctions, for example, and we'll talk about those in the context of particular cell types as we go along. Okay, so in the next section, we'll continue our discussion of properties of cells, transport across the cell membrane, and so forth. So join me for part 2 of our discussion of chapter 3 next time.